stomach. Okay. So when you are drawing the stomach, you should draw in this way. Okay. So this upper portion, you call it as fundus, body, pylorus, antrum, and then you have here the duodenum, which is the beginning of small intestine. And here you have the sphincter muscles called as lower esophageal sphincters. And here you have pyloric sphincters. Okay, so this is the arrangement, how this is the parts of the stomach. And when you see the inner walls of the stomach, you have multiple foldings, what you called as rugae. The walls of the stomach have the foldings called as rugae, which is giving increased surface area availability for the food to be present, because stomach is the place where the food is going to present for nearly 2 to 3 hours. Okay, immediately as like the food bolus entering from oral cavity into the stomach will not release the food whatever you are eating. If it is happening place, always we will be in the toilet continuously excreting, but it is not happening. Once when you take your food for the next 2 to 3 hours, we are not feeling hungry. It means the stomach is still having the food for next 2 to 3 hours, because it takes time to digest. It will not digest all the substances on the whole. To the smaller extent, it will digest and then it will send it to the duodenum. Okay, because if it is not happening in that way, the stomach is not storing the food for a nearly 2 to 3 paracetamol. At least how long it will take time to work out? You're having fever, you're taking paracetamol, and how long it will take time to give the response? At least, what is the half-life of paracetamol? It's your problem. You have to remember all those things. Okay, so it takes time for the food particle, whatever you are eating, it should go and store in the stomach for next 2 to 3 hours. But in case of medicines, it is having its dissolving property. Immediately within 10 to 15 minutes and within half an hour, you will get the response. Okay. And then once when the food enters into the small intestine, that is the place where the absorption of the nutrients from the food is going to take place. Until the small intestine, there is no absorption, only the digestion process is taking place. In the stomach, there is no mechanism of absorption, only the digestion, because the compound molecules has to be broken down into simpler molecules. Okay, so now going into the stomach. So what are the major functions the stomach is doing? The first and foremost function is storage. The storage is being helped by the presence of rugae. Rugae or the multiple foldings which is present in the stomach. So you have, when you see the inner wall of stomach, you have like this foldings. So it is giving more and more of surface area availability for the food to be stored. Okay, so first function is storage, and the second function is mechanical movement. Mechanical movement is the stomach will do the movements like this is the part of the stomach. First this part will contract, this part will enlarge, then this part will contract, this part will enlarge, then the next part will contract, it will enlarge. So now whatever segment which has contracted, later on this will relax and this will undergo contraction. So this kind of mechanical mixing of food will take place within the stomach. So this makes the food to be completely mixed with the gastric secretions. Okay, so this mechanical mixing further helps in the digestion process. Okay, so this makes the stomach to be exposed with all the food particles with the gastric juices. And then third one, the stomach is involved in the secretion of a major digestive juice called as HCl. And fourth one is, this HCl is acting as a disinfectant. And the other one is, it is having a major role in hemopoietic function. Why? How it is taking part a role in hemopoietic function? Because of the presence of vitamin B12, intrinsic factor. This is mainly required for the erythropoiesis formation. Okay, so this is secreted by the cells of the stomach, which you call as parietal cells. Parietal cells is the cell which is giving secretion to HCl as well as intrinsic factor of Castile. And finally, the esophagus, pyloric sphincters, everything which is present, it controls the exit of food always. It is having the duration to excrete the food from stomach into the duodenum. So these are the major functions of the stomach. Now we will move on to the specific cells which is present in the walls of the stomach and what kind of secretions it is going to give. This is very important concept. You have to remember which cell is giving what secretion. Okay, all together so far we are talking gastric juice, gastric juice, gastric juice. But gastric juice comprises of what? How it is going to be secreted. So again, draw the stomach. Okay, so in the stomach you have the, you will segment, what is this upper part? Fundus. This is body, and there you have the pyloric sphincters. Okay, so the cells is having its specific location. Gastric cells giving its secretion is not present in all the segments of the stomach. Some cells is present in the body, some is in the pyloric region. So as the way, the first one which we are going to see is the parietal cells. Okay, so this parietal cell is going to give two major content. One is the HCl and the other one is intrinsic factor. Okay, so parietal cell, it is one of the important cell which is present in the body of the stomach. Okay, it is coming under the oxyntic glands. Just remember this, we have so many glands in the stomach itself. They are involved in exocrine function. Okay, but among that oxyntic glands, okay, under the oxyntic glands you have the division of parietal cells. So this parietal cells is the one which is giving secretion to the HCl and the intrinsic factor. And the other one is, which is also in the body of the stomach, they are called as chief cells. It is going to secrete pepsinogen, which is the one going to be involved in the digestion of proteins mainly. Okay, but this pepsinogen is an inactive form. It has to be converted into pepsin. How? that will take place in the presence of HCl. This pepsinogen will act with the HCl, which is also present in the stomach, and then it is converted into its active form. Will this reaction of conversion activation takes place always? Is it happening always? Always HCl is present in the stomach and pepsinogen is also present in the stomach. Will the pepsin activation happens always? Why? Why no? When the food what you are eating is containing more and more of proteins, because this pepsin is required only for protein digestion. If you are taking highly proteinaceous content of food, then you will get enormous conversion of pepsinogen into pepsin. Otherwise, only to the required amount. Even though HCl is present, if the protein stimulus is reaching your GIT, the gastric region, then only the conversion will take place. So this is the second thing. You are not going to see all the cells which is accumulated in the stomach, only the important cells. And now moving on to the pyloric region. In the pyloric region, you have the cell which is called as G cells. As the name G cells, it is going to give the secretion of gastrin. Okay. And another one you have is the mucus cells, which is going to release rich of mucus substance, which coats the entire wall of the stomach. Okay, here. So the cell arrangement is different. Some cell is present in the body, some is in the pylorus, but it doesn't mean that only those pyloric region have mucus, only the pyloric region have gastrin, only the body has HCl. It is not like that. After the secretion, it will be present all over the stomach. But the cell which is presenting, which is secreting the substances will be present only in these regions. So for example, if you are finding the parietal cell in the histology, then it means you are viewing the body of the stomach. So based on the arrangement of the cell, you will identify which segment of stomach you are going to, you are viewing in the histological image. So this arrangement will help you to identify the part of stomach which is involved in the clinical diagnosis. Pathologically, this is more important to know the localization where the cell is present in the stomach. Okay, so this is the arrangement of the cells, and this thing is very, very, very important. You have to consider, remember the cells which is secreting, parietal cell is secreting what, chief cell is secreting what, and gastrin is secreted by what cell. So this is very, 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 very important. Okay, so you will get at least one MCQ in this concept. Which cell, where it is present, what it is secreting. Okay, clear? The other things you can easily remember if you understand the concept. But this you have to memorize. And then next, moving on to the composition of the gastric juice. So in the composition of gastric juice, again you have water and solids. The same representation, 99.5% and 0.5%. And the solids again divided into inorganic with the same electrolytes, and organic. This will have the difference between the composition of saliva. So what are all the secretions we have seen now? secretion cells and the secretions we have seen just now. Pepsin. And you have lipase. This is called as gastric lipase. In other case, we have seen the lipase. Where? Saliva. That is called as lingual lipase. This is called as gastric lipase. And then next you have mucus and intrinsic factor. So these are the organic substances which is present in the gastric juice. This is the only thing which is having the difference from the salivary juice. And among the organic substances, you have one exception of HCl, which is not seen in the salivary inorganic composition. Okay, next. Now we are going to see the mechanism of HCl secretion. Which cell is secreting HCl? So now we are going to draw the parietal cell. This is the parietal cell of the body of the stomach. So how HCl secretion is taking place? Do you remember the chloride shift mechanism which is seen in the carbon dioxide transport? Carbon dioxide combining with, okay, see over here, you draw the channels which is present in the parietal cell and label the sides. So this part is the lumen. In the sense, if you are giving the stomach, and this is the parietal cell, this is the lumen. And this is the place where you have the blood. So this is what now we are seeing a single parietal cell over here. So this is the lumen where you have the gastric juice. And this other side you have the blood vessel. Okay, clear. So now this is having, the parietal cell is having here the potassium H plus exchange in the presence of ATP. And here you have another channel which is going to transport sodium in exchange of potassium, and you have one more channel which is going to exchange bicarbonate in the exchange of chloride ion. Okay, clear? So these are the membrane transport channels which is present in all the cells. Whether it is going to get activated or not, it depends. Okay. So generally, all the cells is doing its metabolic function. It is getting the oxygen, doing the metabolic function. The function depends upon the cell what we are talking. Okay. And after the function, you will get the end product of the function as carbon dioxide. You will get the end product of the function as a carbon dioxide. So this carbon dioxide, what it is going to do is, so this carbon dioxide, it is going to join with the water molecule which is present in the cell. Okay. And it will give the reversible form of a molecule called as H2CO3. What is this? Carbonic acid. This carbonic acid is very unstable form of a compound. So it will easily get converted into bicarbonate plus H plus. Okay. So this carbonic acid is very unstable form. So it will be easily converted into H plus plus bicarbonate. Okay. So this will happen continuously. Okay. So next what happens? This bicarbonate is sent out from the parietal cell into the extracellular fluid. But this is a negative ion. Bicarbonate is a negative ion. If this bicarbonate is keep on moving out of the cell, you will have the imbalanced equilibrium. So in order to maintain the balance, the bicarbonate is going out with the entry of chloride ion. Okay, and this chloride ion will go and join with the H plus which you got from the splitting of carbonic acid into H plus and bicarbonate. This will not join inside the cell. Okay, this H plus is sent out from the parietal cell into the lumen of the GIT with the help of potassium H plus exchanger. So this potassium H plus exchanger makes the H plus to go into the lumen, and this chloride molecule through the leaky channels, it will go into the lumen of the stomach. In the lumen of the stomach, this chloride and H plus will join and give you hydrochloric acid. So this is how the mechanism of HCl secretion is taking place in the stomach. Okay, clear? So this is what is the mechanism which is taking place. And now we are going to see the specific factor which stimulates the HCl secretion. With stimulating and inhibiting the HCl secretion. Since HCl is having a major role in the digestion, we are considering it into of higher importance. So now just consider, we have three scenarios which is going to happen. First is through the vagus nerve, and next is through the gastric cells, and next is for the enterochromaffins like cells. Okay, so this vagus nerve, it is going to give the stimulus through the acetylcholine. Okay, and now this will go and act on the receptors, and then next G cells which is going to release gastrin, that is also going to act on the membrane, and enterochromaffin like cells, it is going to release histamine which is one of the irritant, it is also going to act on the membrane. Okay, so now acetylcholine, gastrin, histamine, we have three factors which are going to do the mechanism of regulation. Whether it is going to inhibit or stimulate, we will see. So once when they are the ligands, once when the ligand reaches the membrane, it results in the release of second messengers. And this acetylcholine, it is going to use calcium as a second messenger. Gastrin also uses calcium as a second messenger. And this histamine uses CAMP, cyclic AMP, as a second messenger. So finally, what will happen? All these three is going to do its function by activating protein kinase, and this protein kinase is the one which is going to be involved in the stimulation or inhibition of potassium H plus exchanger. Here we have seen this potassium H plus exchanger. This pump is going to be controlled by these factors. If the H plus is going to be entering into the lumen or not, it will be determined based upon these three factors. You will not just like that open the potassium H plus exchanger, H plus is going to release. It will not happen always. It is having this regulatory mechanism. So acetylcholine is going to be released from the vagus nerve. It is a neurotransmitter. And G cells through the gastrin it is going to control. And then histamine from the enterochromaffin-like cells it is going to control. So what are all the receptors we have seen? Here you have the receptor for ACH. They are called as, what kind of receptor it is? Muscarinic receptors. And for gastrin, you have a specific receptor that is called as gastrin receptors. And for histamine, you have another receptor that is specifically termed as hydrogen receptors. And finally, here you have a significance of receptor to activate the ATP which is present in this pump. This ATP has to be converted into ATP, should split into ADP to release the energy molecule. So that is just the H plus potassium exchanger. Okay. So now this 4 regions, 1, 2, 3, 4 regions is going to be controlled by the blockers. So these are all the receptors which is present. So which one is going to block which receptor? We will see now. Muscarinic receptor in the presence of atropine. These are all the blockers which we are seeing now. Muscarinic receptors is mainly blocked by the atropine, and the gastrin receptors is mainly blocked by the substance called as proglumide, and hydrogen receptors mainly by the cimetidine, and finally the fourth one, potassium H plus exchanger, it is by the omeprazole. Okay, so why we are considering this much for HCl stimulation and inhibition is because if a person is coming and complaining of peptic ulcer, you are going to prescribe what kind of drugs? Antacids. Then only antacids? Proton pump inhibitors. What is that proton pump? Proton is this H plus. Proton pump inhibitor. It is going to prevent the pump. This is the proton pump. You call proton as H plus also. Yes or no? This proton pump inhibitor, as the name, it is going to inhibit the release of HCl into the lumen. HCl is formed in the lumen, not in the parietal cell. If the cell is not releasing the HCl, just chloride will be in circulation. No H plus for this chloride to go and join and forming the HCl. Peptic ulcer, there is a erosion in the inner walls of GIT. If the person still has secretion of HCl, it will further cause the erosion and increases the severity. Okay, until the erosion gets healed, you will give antacid to coat the inner wall of the lumen to protect it from further erosion by the HCl. On the other hand, you should also inhibit the HCl formation further in excess. So that is why we are giving proton pump inhibitors. And we will also give hydrogen receptor blockers. Okay. So why this hydrogen receptor blockers? This histamine, it is acting on this is the parietal cell. This is acting on the parietal cell through the hydrogen receptors. If you block this, then the cyclic AMP will not give signal to release the H plus. So this is the indirect way in inhibiting the HCl secretion. So how the peptic ulcer disease is being treated, how the HCl secretion is being inhibited. To understand that pharmacological mechanism, you should know the factors which is inhibiting and stimulating the HCl secretion. Okay. So this is what is the thing which is happening in the HCl secretion, and we will have a break with this. Okay, so regulation of gastric secretion. So we have seen individually how the HCl secretion is being regulated. Okay, so if you want it to be secreted in an increased way, the acetylcholine, gastrin, histamine, everything is taking part its role. Acetylcholine, gastrin, histamine. If you want to block, you have to block these receptors. So this is the action of the drugs what we are giving to control the gastric secretions. So those drugs are acting on these receptors. So it is indirectly helping in reducing the HCl secretion. On the overall gastric secretion, it includes all the composition of the gastric juice, gastrin, pepsin, HCl, everything. Okay, so in salivary secretion, we have seen three phases: cephalic phase, gastric phase, intestinal phase. And as like the same here also in gastric juice secretion, we have the same three phases: cephalic phase, gastric phase, and intestinal phase. So here what is going to happen is, now first we will see the cephalic phase. What is meant by cephalic phase? There is no presence of food in the mouth. You are just thinking about the food, or you are getting the smell of the food, or you are viewing the food. So that is going to give some active secretion of gastric juice. So how that is going to give the signal? So in the cephalic phase, once when the receptors of smell, vision, or thought of the food, those receptors are getting activated, and these receptors are going to give signal to the cortex and the hypothalamus. So immediately once when these receptors are getting stimulated, it will give signal to the medulla. Okay, so from the medulla through the vagus nerve, parasympathetic innervation. So the walls of the GIT is having both enteric and autonomic nerve supply. So here parasympathetic is rest and digest, whereas sympathetic is flight and fight response. So the main thing which is going to be involved from the autonomic nervous system is parasympathetic supply. So its role is to take rest and digest the food what you are eating. So the vagus nerve is getting the input from the parasympathetic innervation, and this vagus nerve is giving the stimulus to the stomach to increase the secretion. So this is how the cephalic phase is giving the gastric secretion, but its concentration is very minimal. And now coming to the gastric phase. So what is gastric phase? The presence of food in the stomach. When you have food in the stomach, the first thing is, once when the bolus enters from crossing the lower esophageal sphincters, then the food is now accumulated in the stomach. It distends the wall of the stomach. The distension of the stomach wall induces the stretch receptors. Yes or no? So now the distension of stomach, because of the presence of food in the stomach, it increases the stimulus from the stretch receptors, and these stretch receptors is now going to stimulate the enteric nervous system, the Meesner's plexus and the myenteric plexus. So once when these are stimulated, that results in the secretion of gastric juices. Okay, so this will be of higher strength than that of the cephalic phase. And the third one is intestinal phase. So in the intestinal phase, the bolus now entered into the intestine. Okay, so whatever has distended, now it is going back to its original position. Stomach is an elastic organ. Okay, It will distend when there is a food, and then it will go back, regain its original structure after the food cross the into the intestine. Okay, so this intestinal phase will get the secretion because of the stretch receptors, same stretch receptors which is present in the duodenum. Okay, so these stretch receptors will give signal mainly through the, once when the food enters into the duodenum will start releasing cholecystokinin and secretin. Okay, so this cholecystokinin and secretin will give stimulus to the gastric region to release gastric juice. Okay, so this is how the intestinal phase is also involved in the gastric juice secretion. But overall, the maximum secretion will be in the gastric phase, because it is the place where the active contact of the food with the stomach is taking place. Okay, clear? So this is the regulation of the gastric juices. And now we will move on to the next concept. Some clinical aspects with respect to the abnormal secretions. So we have the condition called as gastritis. What is gastritis? Inflammation of the gastric mucosa and gastric atrophy. So there is shrinkage in the walls of the stomach if there is any loss of nervous innervation to the muscles of the stomach. Okay. And third one, most commonly encountered clinical scenario, peptic ulcer, which is also called as Zollinger-Ellison syndrome. And the fourth one, when the bolus is not moving from stomach into the duodenum, everything will be accumulated in the stomach. It will be dumped in the stomach because of the absence of opening of the pyloric sphincters, that you call as dumping syndrome. So in this place, the process of gastric emptying is not happening. Okay, clear? So this is the clinical aspect scenario with respect to the stomach. And now the stomach, the role of digestion in the stomach is over. Now it has to move the bolus from stomach into the duodenum. So we have to see the kind of movements. From the oral cavity into the stomach, it is by the process of deglutition. And now from stomach into the duodenum, what are all the patterns of movements the stomach is undergoing? Movements of stomach. Okay. So in the movements of stomach, first movement during empty stomach, that is before meals. So this you term it as hunger contractions. So this type of hunger contraction, if it is once started, it will go up to the ileum. Okay, so this kind of movement is mainly to clear out the residues from the previous meal. Okay, so this is the movement before the intake of meal. Okay, so they are called as hunger contractions. And the next one is movement after food intake. So in this case, you have receptive relaxation, that is what we are terming as the distension or stretching of the stomach. Okay, so it is stretching and receptive relaxation is taking place. And next one is the mixing waves, which I have already drawn and show you the picture how the mixing is taking place. If one segment of stomach is contracting, the other segment is relaxed. Then the contracted segment will relax and the relaxed segment will contract it. So this is how the contents of the GIT is mixing all over the stomach. Okay, so that is called as mixing movements. So these are the major movements which is helping in the gastric emptying. The normal gastric emptying duration is 2 to 3 hours. Okay, here. So up to this, the digestive role of the stomach is over.